sooner we start, the sooner we'll finish. Let's start. So I clap my hands. Okay, so the second part of this talk will be synaptic models. I'll try and go very fast, so I'll skip lots of slides here. But as I said, I'll leave all the slides for you to read. So the idea, since this is a school, I put a lot of information here. I knew that I have more material here than I would be able to cover in, say, 3-hour talks. But that's on purpose, because I'll give you lots of information, and then you can using this information, you can study at home and find other material to learn about this. Okay? So that's the idea of having this. So what's the purpose of synaptic modeling? So it's to capture the following facts. That some neurons have stronger and more lasting influences over a given neuron than others. So one wants to model this in a network model. Some of these influences are excitatory, which means that they increase the likelihood of a spike emission by a cell, while some are inhibitory. They decrease this likelihood. So you want also to capture this phenomenon, excitation and inhibition, and the strength of the influence of a neuron over another one changes over time as a function of the activities of both neurons. This is synaptic plasticity, as I mentioned to you yesterday. So you want to capture all these three phenomena when you want to model synapses in a network model. So there are two types of synapses in the brain, chemical synapses and electrical synapses. Electrical synapses are basically two neurons coupled by a junction, an electric junction, which is called gap junction. It allows rapid communication between the cells because material can flow from one cell to the other in both directions. So it's bidirectional. It allows bidirectional communication. So it's not only on one way, but it's both ways. Can be either excitatory or inhibitory at the same synapse because it depends on what flows from one cell to the other. So the gap junctions, electrical synapses can be excitatory or inhibitory. And it also occurs between neurons and glia. So if you want to model glia, and you want to model coupling between neurons and glia, you have to use gap junctions. Chemical synapses, well, they do signal transduction, which means that they transfer the input signal into different output signal. They could be either excitatory or inhibitory, but not both at the same time. They are slower than gap junctions. They are unidirectional, so it goes only from, say, presynaptic neuron to postsynaptic neuron. If you have two cells, A and B, one of them should be the presynaptic one, and the other one should be the postsynaptic. If you want to have a synapse from B and A, this would have to be conveyed via different synapse. And they are more plastic, and this is probably the most important feature of the chemical synapses and why they are so important, because they can change. And the gap junctions are more or less fixed. The conductance, the effectiveness of the synapse is fixed. So that's why they're fast, but they're not plastic, and this really is an important feature. So electrical synapses, I mean, since they're due to the gap junctions, which are proteins that called connectines that connect the cells, they can be easily modeled just by putting an axial resistance joining the two intracellular media, the intracellular medium of the cell 1 and the intracellular medium of cell 2, and then you couple them via a resistance or a conductance with a given value. So the voltage of one of the cells is related to the voltage of the other cell via this simple equation here, and the current that flows along the gap junction is given by this equation here. So it depends on the difference of values between the two voltages. If the voltages of the two cells are the same, so they are isopotential, so there's no current flowing. So they can be modeled by a single resistance connecting the two cells. So it's very easy to model a gap junction. Chemical synapses. Oh, a chemical synapse is a very complicated thing. Basically, when the nerve impulse reaches the terminal of a presynaptic cell, it depolarizes this region, and this depolarization opens calcium channels through which calcium ions flow inside the cell, the presynaptic cell. They diffuse inside the button, the synaptic button here. They bind to vesicles that contain neurotransmitters, and this binding drive them towards the membrane of the presynaptic cell. When they reach the membrane, they fuse in the membrane, so they break and they release neurotransmitters in the synaptic cleft. The neurotransmitters diffuse through the synaptic cleft, and they bind to receptors in the postsynaptic cell. And the binding can open the receptors, and the receptors are ion channels, so they lead, say, ions, say sodium, to enter the cell, and the entrance of the sodium can depolarize the cell, creating a postsynaptic potential. Could also, they could be, say, potassium channels, and potassium can exit the cell, and these hyperpolarize the current. And there are also receptors that do not open, but they release second messengers inside the postsynaptic cell, and the second messengers create a cascade of events that eventually will open some ion channels on the postsynaptic cell, and this would lead to depolarization and hyperpolarization of the cell. And this would eventually create nerve impulses.
I'm sorry, I saw this model, let's say model about the...
This is a very simplified scheme.
No,
yeah.
But I have some difficulty to understand the velocity of all of those processes. When you measure the velocity, for example, the ions that are coming from one side of the neuron and going to the other side of the neuron, is that possible to measure this velocity?
Yeah, it is.
This vesicle must travel, let's say, not using ground and motion more or less to the other side of the
Yeah, it's diffusion. Basically, it's a diffusion process. You can model this. Yeah. Yeah, lots of several processes. Yeah, it's possible to model all of them. You have, I mean, people from this school here, I think, have all the technology to do that model. So you can model single synapse, and then you can model many of them in a network. So you can do that. But then, of course, the cost of simulating all of this is really, I mean, very high. It's possible to have a network model with thousands of cells, with millions of synapses, because the number of synapses per cell is, say, 10 times. Say one typical cell makes 10,000 synapses. So 10 times the number of cells. And then you can do that. But it's really expensive. You know? There's this joke. I cannot resist. I have a friend who used to teach at Cold Spring Harbor, a Brazilian friend who used to teach there the computational science. And one day he was walking in the sidewalk there, and he found some tourists, some American tourists. And they asked, "Oh, you're not from here. What do you do here?" "Oh, I teach computational neuroscience in the school here." "Oh, so you work on the brain?" "Yes." "Oh, we're very interested on the brain. So explain us how the brain works." I said, "No, I only work on the visual system." "Okay. So explain us, visual system is important, vision is important. So explain us how vision works." "No, I work on the vision of the fly." "No, okay, let's understand the vision of the fly." "No, I work on the H1 neuron of the visual system of the fly." "Oh, okay, so explain." "Oh, no, I work on the molecule, blah, blah, blah of the... That's the problem with biology. So you can understand the molecule, but it's difficult to understand. So anyway, so a chemical synapse could be either excitatory or inhibitory. It could be inotropic, which means fast, or metabotropic, which is slow. So the inotropic ones is the one that's related to the opening of the receptor gate, and that allow the flux of ion channels. The metabotropic ones are the ones related to second messengers that go through a cascade of sequence of events inside the postsynaptic cell and eventually open a receptor. The most common neurotransmitter is in the brain, in the cortex. I'm focusing on the cortex here. Glutamate, which is usually excitatory. So the major excitatory neurotransmitter in the brain is glutamate, and gamma-aminobutyric acid, GABA for short, which is usually inhibitory, and is the major neurotransmitter, inhibitory neurotransmitter in the brain. And the receptor types are mainly AMPA or kainate or NMDA, which are the glutamate receptors, and GABA-A and GABA-B, so-called GABA-A and GABA-B for GABA. So those are the receptors. And since they are plastic, they're prone to short-term and long-term synaptic plasticity phenomena. So postsynaptic potentials are just the potentials generated on the postsynaptic cell due to a synapse. So this is in the presynaptic cell, you have a spike. When the spike reaches the synaptic button, it generates a fluctuation of the voltage in the postsynaptic cell, which could be positive, meaning that the postsynaptic cell was depolarized. So in this case, you have an excitatory postsynaptic potential, EPSP. That's the jargon for the postsynaptic potentials that could have different shapes. But in general, they're all subthreshold. Or they could inhibit the postsynaptic cell. They could hyperpolarize the cell. And in that case, you have an inhibitory postsynaptic potential, IPSP. So EPSP for excitation, IPSP for inhibition. A single postsynaptic potential, excitatory postsynaptic potential, cannot generate a spike, but a series of them can generate a spike because they propagate through the dendrites to the soma, and then they add that. So you have two types of summation, temporal summation and spatial summation. So temporal summation is when you have a sequence of postsynaptic potentials from different synapses, and they add up. And then if the temporal summation crosses threshold, you have a spike. Or you can have several synapses from different neurons, and they add spatially. They add on space at the same time. And if the summation of them also crosses threshold, you have a spike. Here, a cartoon for ionotropic and metabotropic synapses. Synaptic receptors, as I said, the ionotropic ones are AMPA and NMDA. AMPA is related to so-called early EPSP, so the early depolarization of the cell. NMDA is lower because the NMDA channels are blocked by a magnesium ion. And the magnesium ion is released only when the cell is depolarized. So NMDA channels to be open, they require a previous depolarization of the cell, so a previous excitation of the cell via AMPA receptors, and then the magnesium ion is unblocked, is released, and then the NMDA channels open, so they're even slower than the AMPA channels. And the GABA A is ionotropic, and GABA B is metabotropic, so they require lots of chemical reactions, biochemical reactions inside the postsynaptic cell. So how to model then? So there are many models, but usually, since eventually they lead to opening of ion channels on synaptic channels, which are ion channels on the postsynaptic cell, you can model then using the Hodgkin-Huxley formalism, the same formalism that one had before, which means that you have a synaptic current that is given by a synaptic conductance times the difference between the membrane potential or potential of the postsynaptic cell minus the equilibrium value of the voltage for that channel, which is not due to a single ion in this case, because the synaptic channel can be permeable, can there be fluxes through it from different cells, from different ions. So people refer to this equilibrium voltage as reversal voltage or reversal potential, because whenever the voltage crosses this reversal potential, the current changes sign. So, but the formalism is the same. The reversal potential for inhibitory synapses is about minus 75 millivolts, so they hyperpolarize. It's the same mechanism of the ion channels I explained before. So they act as driving forces. They force the channel, the conductance of the current, they force the voltage to go to the equilibrium value of the type, the synaptic type. So if it's inhibitory, usually it inhibits, it hyperpolarizes the cell. If it's excitatory, the reversal potential is positive. Usually people assume 0 as the value. So they tend to take the voltage to depolarize values. And what about the synaptic conductance? I'll talk about that in the next slide. And then you can also add some delays, because you have the travel time from the presynaptic cell to the postsynaptic cell. So you can add a synaptic delay time. So whenever a presynaptic cell spikes, you only introduce a change in the conductance of the postsynaptic cell after a certain delay time. So you have to add that in your model. So about the synaptic conductance. So you have to, basically what you do is you have to model the time course of the postsynaptic potential, which follows the time course of the postsynaptic conductance. They can be modeled by kinetic equations or molecular biology, as some students asked me. So you can use whatever tools you want to model these. But if you really want to simulate lots of cells, you want to simplify this step. Since you know that most of the postsynaptic cells are more or less stereotyped, or say they are due to AMPA or to GABA or NMDA, they more or less have the same shape and time course, same temporal characteristics. What usually one does is to use a fixed function to model the time course of the conductance, of G of t. And then you have, based on several experiments, the typical time course for AMPA conductances, NMDA conductances, and GABA conductances you have here in this graph. So AMPA is by far the fastest one. It's really fast. In this time resolution here is almost like a vertical line. And then NMDA is slower. GABA A is also fast. It's inhibitory. And GABA B is lower because it's a metabotropic receptor. It's mediated by a metabotropic receptor. To model those time courses, one basically has three types of functions. Either you say that there's an instantaneous rise in the conductance followed by an exponential decay, and the exponential decay is just modeled by this equation here. G bar is just the maximal conductance the peak of the conductance there. You can use so-called alpha function, which is a function that has a rise time and decay time. The rise time is not instantaneous, so you capture the rise time, and then the decay time is very popular among models of biophysically detailed ones. And the most, say, detailed one, if you want, is just the sum of two exponentials, which is this equation here, which gives you this behavior here. So you can choose one of them. In general, for network models that use simple cells like leaf or two-dimensional leaf models, people use this one here, instantaneous rise plus decay. So they have a single parameter, actually two parameters, which is the maximal value of the conductance, of the synaptic conductance, and the time constant for that. But for people who model biophysically detailed single cell models or network models, they use either alpha functions or this combination of exponential functions that require more parameters. Say, for example, here, synaptic inputs to a leaf model, as I said, also valid for other integrated and fire models. You can have two possibilities, two scenarios, either current-based or conductance-based. If it's current-based, you add current to the voltage equation, and the current is described by a fixed function. So you directly model the synaptic current generated by the synapse into that cell. Or you can have a conductance-based scenario in which you have basically the same equation, but now the synaptic current here depends on the synaptic conductance. And the synaptic conductance is modeled by a fixed function. So you model the synaptic conductance first, and then you multiply by a term here which would capture the efficacy of that synapse, and then you have your synaptic current, which you plug into that equation. So this has an extra step. You model the conductance first, and then you model the current based on the conductance. So this one is a little bit costlier than the other one. Synapses also, they have two types of plasticity, long-term and short-term plasticity. Short-term plasticity is a phenomenon that is observed experimentally whenever you generate a train of spikes with a given frequency in the presynaptic spike, in the presynaptic cell. The postsynaptic cell, after a certain time, it responds with a higher amplitude postsynaptic potential. So the amplitude of the postsynaptic potential can increase for the same input in the presynaptic cell after a train of excitations of that cell. So you see the amplitude here increases up to a certain frequency. After that frequency, you have depression. So you can understand that, assume that you have depletion of neurotransmitters. So the amplitude of the postsynaptic response decays. So you have facilitation and depression. These are short-term plasticity because they occur in short time scales. They don't last for longer. because they're variable. One can model this by introducing in your conductance model, which was previously just the maximal conductance times the fraction of the probability that the channel is open. Then you add another term, P relative, which measures the probability that a transmitter is released by a presynaptic neuron following the arrival of an action potential, and depends on the frequency of the presynaptic cell. So these are phenomenological equations, so they don't consider molecular biology. But of course, you can use different models to capture the same phenomenon, facilitation and depression, but this is the simplest one used in popular models. So you just have two equations there, one for facilitation, this is the frequency for facilitation, one for depression. So you can use this in your model. This is a simulation, for example. And then you have activity-dependent synaptic plasticity, which is believed to be the basic phenomenon underlying memory and learning. As I said yesterday, Hebb was the first one to propose a model for that, which was model based on words. He didn't do any mathematics. Initially, his suggestion was only for strengthening synapses. More recently, the model, Hebbian models have been generalized to include also synapse models in which the synaptic strength decreases due to the failure of a presynaptic cell in exciting the postsynaptic cell. And in the 70s, people found experimentally that some possible biophysical candidates to Hebbian synapses, so-called long-term potentiation and long-term depression. They initially were found in cells of the hippocampus. Nowadays, they have been found everywhere in the brain. So you can have LTP and LTD basically everywhere in the brain. Basically, it's assumed that any synapse in the brain is possible to exhibit, to display both LTP and LTD. So what is LTD? LTP. If you stimulate the presynaptic neuron for a short time with a high frequency, so you generate a high frequency train of spikes in the presynaptic cell, which leads to depolarization. So you need the response of the postsynaptic cell to this high frequency train of stimuli. After this period, you have potentiation. So a given presynaptic stimulus generates postsynaptic responses which have higher amplitudes. And these last for long times, say minutes, hours. It's called LTP. But then if you fail to stimulate the cell, or you stimulate with very low frequency stimuli, the presynaptic cell for a certain time, you have the opposite effect. The amplitude of the response postsynaptic potential is very low. And this is called LTD. So this protocol, this experimental protocol, is used to test whether synapses display LTP and LTD, and they display both of them in different areas of the brain. The mechanisms underlying LTP and LTD are very, very complicated, so this is a task for you, not for me, because computational neuroscientists are more interested in the behavior of networks than in the behavior of single synapses. You here have a cartoon from different mechanisms involved in LTP and LTD. It's an active area of research. Still, we don't understand all the mechanisms. But anyway, they are very complicated. But the generic mechanism is like that. So a presynaptic cell is stimulated. So the synapse is stimulated, releases neurotransmitters. They bind to receptors. And there might be some mechanism here that increases the number of receptors. And the increase of the number of receptors is followed by the increase of the number of neurotransmitters that are released by the vesicles. And then you have both large number of transmitters and large number of receptors. Then you have a stronger synapse. How does this operate? Nobody knows as yet. There are experiments that show that they are related to memory, because one say, oh, this is just a phenomenon that happens in the brain, but people have done experiments with animals, different animal models, and they blocked LTP or they blocked NMDA because it has been shown experimentally that NMDA receptors are involved in LTP, as well as calcium in the presynaptic cells. And among several other mechanisms, and they block those parameters, and they observe that the animals which have those parameters affected or blocked, they have memory deficits or behavioral deficits that would depend on memory. So there's a direct link between LTP and memory formation. So because of that, so if you want to model this, the favorite type of model is Hebbian plasticity, as I said yesterday, proposed by Hebbian in 1949. So his motivation at the time was to have a model for the creation of cell assemblies, as he used to call at the time. Today, people still use the same terminology, cell assemblies and learning. So neurons that fire together, wire together. So here you have an example similar to that one of Chewbacca that I've shown to you yesterday. So I have a group of cells interlinked via synapses that represents a banana, and another group of cells which can share some elements from the former group that represent an apple. So those would be assemblies in the brain that would represent different elements of the external world that you mentioned, that you want to memorize. Much more recently, in the late 90s, Macron in Switzerland and other colleagues, they found this phenomenon called spike time independent plasticity, which is a causal version of the Hebbian rule. Because Hebbian rule is solely based on the correlation between the two cells, pre- and postsynaptic cells. It doesn't care about the timing of the presynaptic and postsynaptic spikes. But if you think about a little bit about it, you ought to have a causality there. So the presynaptic cells, if the presynaptic cell influences the postsynaptic cell, the spike in the presynaptic cell has to precede the spike of the postsynaptic cell. If it's the other way around, there's no causal link between them. So there's just a spurious correlation between the two of them. So you must have the presynaptic cell firing before a short time interval before the postsynaptic cell. And they found this experimentally, and this was called STDP, spike time independent plasticity. So there's LTP, long-term potentiation, if the presynaptic spike occurs before the postsynaptic cell, which corresponds to this part here. So they have plots like that. No? And W would be the synaptic strength. So if the pre occurs before the post, you have the strengthening of the synapse. But if the pre occurs after the post, so it's just a casual spiking, you have L to D. So these dots here are experimental data, and you can model this. There are different STDP models in the market nowadays, so you can choose one. And this has to occur within a short time interval. If the pre and post have any relation outside this time interval, they don't affect each other. So the timing has to occur within a short time interval after the presynaptic spike. So this is STDP, and there's a whole industry of modeling of this phenomenon at the moment. So if you want to model a long-term plasticity, using, for example, a Hebbian mechanism, and using the Hodgkin-Huxley type formalism, what you can do, you can increment the G bar. So you don't care about SC and the other parameters because they capture just the time courses of the specific type of synapse you have. But then you want to change the maximal conductance, because the maximal conductance is the parameter that captures that increase of transmitters and receptors in the synapse, which is the maximal value that synaptic conductance can have. And then you need to have an equation for this, which has a time course which is much slower than the time course of the neuronal dynamics. And then you can implement a Hebbian type rule. So, for example, if I have a rate-based model, so a model using rate-based neurons, this is the equation that I have. So the output of the neuron is just a function of the input, and the input is just the product of the input the neuron receives times the synaptic weights. And then how do you change the synaptic weights? You change in a Hebbian way. So you make the change of any particular synaptic weight proportional to the product of the firing rate. Remember, in a rate-based neuron, the variables are the firing rates. So the model naturally can include firing rate-based mechanisms like the original Hebbian rule. So Xi is the firing rate of the, say, neuron 1, or the ith neuron, and S is the firing rate of the postsynaptic neuron. So you just make the product of the two of them, and then you increase the synaptic weight based on this. So if both of them are firing a high frequency, you increase the weight. Of course, you need to add some extra terms there to limit the growth of the synaptic waves. Initially, the first models that used this equation here, they had overflow of the variables there because they didn't care about these. But then they learned afterwards that it's important to limit the growth of the synaptic wave because it has to do to the number of transmitters and receptors and so on. Another popular model is so-called BCM model, is Bienenstock, Cooper, and Munro. This is the same Cooper from Bardeen, Cooper, and Schriff, yeah. BCS from physics, and so they had the BCM model. It was introduced in 1981. So it's also a Hebbian type model. So X is the presynaptic activity, firing rate of the presynaptic cell. Y is the postsynaptic firing rate. W is the synaptic weight. And then you change the synaptic weight based on a function of the postsynaptic activity times the presynaptic activity. And there's a decay term here, which is added just to limit the growth of the synaptic wave. And the novelty is that this function here has this shape here. And it has a threshold here that can be adjusted. So you see this function here, which was proposed in 1981, it more or less at the same time that LTP and LTD were being discovered, it can capture the two phenomena because you can have a depression phase here and a facilitation phase there, a potentiation phase there. So people use BCM models to try and model both LTD and LTP. Much later by, I think, Izikevich, it was proved that under some conditions, this model here, the BCM model, can be derived from an STDP model. So one can link those models. So this is a model that is used as well. So I think that's the stopping point for this lecture, for the synaptic. So I just gave you some ideas of how to model synapses. Let's move instantaneously to the third lecture, which now starts with the part 4 with network architecture. So so far we have seen single neuron models, synaptic models, more or less a broad overview of those neurons, of those models. And then you want to construct a network model. So you want to assemble your neural models connected via your synaptic models into network models. So what type of network model or network architecture you're going to choose? So basically you have two options there. You can either choose an artificial architecture, which is an architecture which is constructed by hand, or you can use a data-driven architecture, which means an architecture which is given by experiments, by data, so biologically inspired. If you want to choose an artificial architecture, you have freedom to choose over several models. There's a whole industry of so-called complex networks at the moment in the market going on in science. So you have lots of different possible network models, graph models in mathematical terminology that you can choose from, stemming from, the regulum in which, a given neuron, a given point is connected only to nearest neighbors, to completely random network in which each pair of neurons is coupled with a given probability p. So you can have both short or long-term connections there. I'm sure you know about complex networks, no? So you know how to model that. And you can have in between the two so-called small world networks in which have both local connections and long-range connections. And there's a whole zoo of complex networks there. You can characterize them based on randomness, as here, on modularity, because you can have some networks that say, we are in a modular structure. We are in a room here amongst other rooms in the same building, and this building, together with the other buildings, is part of UNESCO, and UNESCO is part of the system of universities in the state of São Paulo, which is part of the system of universities in Brazil, and so on. So you have modules within modules that follow a hierarchical structure. So this is the idea of hierarchical modular networks. You can characterize them by heterogeneity, and then you can take the data and classify your network type based on the features here. So you can choose by hand one of those networks and do a model based on this and see what is the behavior of your artificial neural network, artificial network. Or you can choose an architecture that is inspired on biology, on what biology or neuroanatomy tell us. So the idea is to incorporate elements and quantitative information of real network architecture. So you can have cortical-like architectures, architectures based on the visual system, on the olfactory system. You could choose a given area or region of the brain, and you take anatomical data from this area, and then you construct your model based on this. So you have to constrain your model to satisfy parameters or information that are accessible about that brain area. Also, you have to be concerned about the spatial scale of your model. What you want to model with your network model. You want to model a local circuit, or you want to model the entire brain, for example. So if you want to, say, model the entire brain, you need, say, somehow a graph like this adjacent matrix, which is from the macaque cortex. So this is only for the cortex, but from the entire cortex of the animal. So you pass for the cortex into different areas, brain areas, which can be determined by different techniques, and then you estimate how these areas are connected, how many, say, axon bundles connect the areas, and this would give you an idea of the strength, synaptic strength that connects the two areas. And then you'd have a directed graph, a weighted graph, but then you can binarize this graph just by putting a threshold there. So connections that are above a certain threshold are considered as 1, connections that are below are considered to be 0, no connection at all. And then you can construct a network like this. So this would be a network for the entire connectivity of the macaque cortex. All areas corresponding to the part of the brain. Or you can just have a model for, say, you take a cortical column. One of the hypotheses about how the cortex is structured is that the cortex is subdivided into columns, vertical columns that span the entire depth of the cortex, and then you have lots of columns. And the columns would be kind of secondary elements. So the neuron would be the most important element of the brain, and the columns would be the second most important elements of the cortex, at least. And the way the columns are assembled together would give us some hint of how the cortex works. So if you want to model a cortical column, you say 1 or 2 millimeters of surface area, you need to have information about the connectivity within that area. And then you have ways of do that, and then you can construct a network that captures the connectivity of that specific cortical column. You can have models for other brain structures, so like this is for the hippocampus, for the dentate gyrus of the hippocampus. So it's made of different cell types, different connectivity, not layered, as in the cortex, but you have access to this type of data, and then you can construct. So these would be local ones, these would be microscopic, this would be macroscopic. So you have microscopic. This is not my slide. I took from Damian Battaglia, who just gave a course in the school that I organized here in January. So I took some slides from his, because I was lazy to copy. But anyway, it gives you the idea. Damian is based in Marseille, in France. So microscopic would be models that, say, go from a column or to a few columns, say 1 millimeter to 1 to 10 millimeters of area, surface area. So for that models, you need to know all the cells in that region. They would form so-called local microcircuits. But then if you go to macroscopic models, you have models that capture the behavior of the entire brain. And then you need some way of measuring how different areas of the brain or how different structures in the brain are interconnected. And you can you can be helped in this by modern techniques like functional magnetic resonance imaging and tractography that measures the diffusion of water along the axons that connect the cells, and then you can measure the strength of the connectivity between different areas. So just a little bit about microscopic architectures of the cortex. So the cortex, the basic structure of the cortex, this is known since the beginning of neuroscience and the early 20th century. If you stain a tissue, cortical tissue, you see that the cell bodies have different concentrations depending on the depth along the cortical surface. And based on analysis of different this dimension here, the vertical dimension of the cortex, into layers, 6 layers, 1, 2, 3, 4, 5, and 6 layers. So that's the usual classification of the cortex. The cortex is subdivided into 6 layers, apart from the columns. There are horizontally 6 layers. And more recently, there have been several studies that really made a comprehensive, that allowed us to construct a comprehensive scheme involving excitatory and inhibitory cells in various layers, along with external thalamic inputs. So the effort of a whole generation of neuroanatomists, neurophysiologists, and other neuroscientists resulted in the situation that we have now, in which we have access to very detailed information about how those cells in local circuits are connected. What type of cells, if they are, say, have a different firing type, if they're excitatory, if they're inhibitory, their proportions, how they are connected within a given cortical column, both vertically and horizontally, and how they are connected to other cortical columns. So we have nowadays information, very rich information about all these structures. So here are some papers. You can go through those papers that they are based on studies on cat, mouse, and rat, and rodents, and other animals. They use both anatomical and physiological techniques, and they can be condensated in models like this so famous Postsynaptic. They are researchers from Jülich Research Center in Germany. Markus Diesmann is the leader there. So it's a model that combines information from over 50 experimental papers on the connectivity, local connectivity within a given column from mammals. So it's local cortical microcircuit. The surface is 1 square millimeter. It's comprised by 8 cell populations. So it has 1, 2, 3, 4 layers. Layers 2 and 3, they lumped the 2 layers into a single one. Layer 4, layer 5, and layer 6. And in each layer, they have excitatory and inhibitory cell populations. So you have, in the end, 8 cell populations. The total number of cells is 10 to the 5, so it's really a large-scale model. The number of synapses is 10 to the 9. So it's really, really billions of synapses. 80% of them are excitatory, 20% of them are inhibitory. This is the correct proportion of excitatory and inhibitory cells in the brain. Okay, and now for macroscopic architecture. So how you describe the architecture of the whole brain. So the basic technique nowadays, you use some technique like a functional magnetic resonance imaging, fMRI, to measure activity of different brain regions, brain areas, because they're not able, this technique is not able to capture activity of single cells, not even local circuits, but from large populations of cells, which correspond to brain regions. So each region would be a node in your network, and then you link the nodes via, links that are given by the axonal tracing or diffusion tensor imaging. So these are 2 techniques. Tracing is the traditional technique. You inject a dye or a virus in the tissue, and then you see how it propagates, both from an injection point to the synapse that cell makes, or from the synapses backwards to the cell. So they are anterograde and retrograde tracing, and then they allow us to see how far a connection from one given cell reaches. Or you can also measure the diffusion of water along the axons that comprise the brain. DTI, that's the name of the technique, and based on that you can construct images like this, which give you an idea of the number of, these are not real axons, but just traces that connect different areas in the brain. And by counting as I said before, the number of traces, you have an idea of the magnitude of the connection, and you can binarize this, and then you can just have dots that you can put in an adjacency matrix like this one, showing you which regions are strongly connected to other regions. So the names of the regions will be given here. And then you can analyze, using graph theory, the structure of the resulting architecture. So in general, what people have observed, at least for the cortex, is that the general structure is hierarchical and modular. So you have modules that comprise a given hierarchical level and other modules at a different region, and then they are part of another hierarchical level that are part of another hierarchical level, and so on and so forth, as the example I gave about the universities here. And they also have hubs, which are nodes that are very interconnected to other cells, and they found that the hubs, they have the likelihood of being connected to other hubs. So this is called rich club structure. So the rich are connected to the rich. That's why the richer get richer, because they talk to themselves. So here you have the adjacent matrix of the hierarchical modular network, and here, I mean, papers like this will describe this rich club structure to you. So it's possible nowadays to characterize the structure, the connectivity structure of the whole brain, at least of the whole cortex. And then there's the connectome, the famous connectome. So the famous, we're in the age of the ohms, no? The genome, the connectome, the dynome, the metabolome, the whatever ohm. So there's a whole industry of ohms going on. And some model animals are being investigated. And the aim is to map all the cells and connections, to map the entire graph of the brain of those animals. The ultimate goal would be to do that for humans. But the animals that are being investigated at the moment are the C. elegans, Canobilans elegans, something like that, which is a worm, the fruit fly, the mosquito, the butterfly, the mouse, the macaque. So these are the candidate animals. So I took these slides from Eli Schliesselman, who gave a talk or course on the school that I organized, just because, I mean, I didn't have time to transfer them. So C. elegans, this is a model organism for the investigation of animal development. So it's a very simple, one of the simplest nervous systems that one has available is a worm. So it's very small, has only 302 neurons. The genome was mapped in 1998. The somatic connectome was mapped 35 years ago. The synaptic currents as well. So I think it's by far the most well-understood system, nervous system on Earth. Evolutionary information is available. There are lots of information. You can access this information accessing this webpage here, wormatlas.org. So you have there lots of information about the C. elegans. Then you have the fruit fly. The fruit fly is important because it's much more complex than the C. elegans. And the Flyem, this company here, is making the data, is doing this, the construction of the connectome. And also, all the tools necessary for investigating it have been available for free for the community, so you have access to it. And the bioRxiv paper was just published 2 months ago, January 21st, 2020. And they mapped the whole connectome of half of the Drosophila brain. The Drosophila is the fruit fly. And they plan to map the entire connectome of the fruit fly by 2022. In 2 years' time, you have access to this as well. Zebrafish is a small fish, also a very popular animal model for neurobiologists. You have here the pages. This is not the entire connectome, but the connectome of the olfactory bulb. So all the cells of the olfactory bulb and the connections there, the edges that connect the nodes. For the mouse as well, you have a mesoscale connectome, not a full connectome, but a mesoscale. So they lump lots of areas into larger areas, and they connect those areas. This is being done by the Allen Institute, which is based in Seattle. Allen Institute was founded by Paul Allen. I'm not doing any propaganda, but it's one of the private institutes that is investing money into neuroscience research these days, and they are making all the information available for researchers, for theoreticians to model. For the macaque, one has not a full connectome, but one has the connectome for the vision-related cortical areas. These are well known since the 90s, and it's been curated, which means maintained by people from ULISH at the moment. So you can construct a model, an entire model of the visual-related areas of the cortex of the macaque monkey, which is the closest one to us from all the animals I've shown to you here. So that's only for you to have an idea. And there's also the Human Brain Project, which is far-fetched goal, is to have the connectome of the entire human brain with maps and so on. But we're still far from this. But you can access the page. You can just type Human Brain Project and you have access to this information about this project. But then having the connectome is not enough. This is one of the lessons one has. It's not enough to know all the nodes and the edges that comprise a given network. You all know that. It's not enough. So one needs to, because the connectome gives only a view of the static graph of connections there. So this is called a structural connectome. This is another name for the connectome, the structural connectome. One needs to go beyond that. One needs to capture, as I wrote here, somehow the statistical interdependencies among the activities displayed by the network nodes. So you know that the network nodes have activities because they are made of cells. And the connections, the edges themselves, they change because they are plastic. So you have the entire system is dynamic. The nodes are dynamic and the connections are dynamic. And they must have some sort of interrelation. And this would result in so-called functional connectome, how they're functionally connected, how the spike of one region here is correlated. I didn't want to use this word, but that's the word that most people, is correlated to the firing of another cell there. I didn't use the word causally connected because this is, I mean, you know that correlation doesn't imply causality, but I mean, how is at least this correlation? This would give a hint about the functional connectome, the functional connectivity. So you can have, in the same way, same fashion, have a connectivity for the structural connectivity. You can have an adjacency matrix for the functional connectivity. How can you do that? So you take your image, say fMRI image, and then you pass the brain into different areas. And then you can use the different areas and using those techniques that I mentioned to you earlier. So diffusion tensor imaging, axonal tracing, you can construct your structural connectivity. And then you can binarize it and have this connectome here. But you can take the nodes from your brain map and somehow measure the activity of each node, of each region. Say the spiking activity of all the region, or the voltage of the activity, or the blood flow, because that's what fMRI measures. fMRI doesn't measure the spiking activity itself, but you know, when a neuron is highly active, it requires lots of oxygen to be active, and this is brought to it by blood. So fMRI measures the blood flow. And by this, you have a measure of the activity of the cell, of different regions. And then you can correlate the activities using any correlation measure. And then you have a map of the correlations between any pair of areas in the brain. So you have a correlation adjacency matrix. And then you can binarize it in the same way. And then you can create a functional connectivity matrix that captures the degree of functional relationship between different brain areas. So you have structural connectivity maps and you have functional connectivity maps. And they must be somehow related. But how? How to relate a structural matrix or structural graph to a functional graph? A possible way would be to use the so-called dynome, another ohm in the zoo of ohms. So what is the dynome? The dynome would be the connectome plus the neuro and synaptic dynamics. So if you add to your connectome the dynamics of the nodes and the dynamics of the edges, you'd have the dynome, which is a kind of temporal, kind of connectome that evolves in time as the nodes and the connections change themselves, as depicted in this cartoon scheme here. So the connectome is dynamic, both in terms of nodes and in terms of connections. So I also took these slides from Demian. So from structural connectivity, we know that structural connectivity shapes the dynamics. This is similar to a slide I showed to you in the first lecture. And the dynamics also shapes the functional connectivity. If you do computational modeling based on the structural connectivity, you can eventually model the dynamics. You can model the dynome because you have the time evolution of your model. And if you analyze the functional connectivity using different techniques, you can understand how this is related to dynamics as well. So this more or less captured the main problem of computational neuroscience at the moment. Interlink all those aspects here. But there are several questions here. Also, Damien wrote this, but I subscribe. So how to quantify functional interactions? How to change functional connections without changing structural connections? How to keep, so with the same structural basis, how to change functionality in the network? Are the functional dynamics useful for concrete applications? These would be for people who are interested in artificial neural networks, for example. Can we infer structural connectivity from activity of the neurons there? Can we detect complex dynamics in neuronal activity and explain it through models? That's one of the main tasks of computational neuroscience. Can we build models of realistic circuits? Do their structure really matter? So is it really important to know the structure, or just the dynamics or the functional connectivity is important? Okay, so I went to part 4 with these open questions, and I have more or less 15 minutes to finish. So I'll put everything together and give you some examples of models that I construct with collaborators. So models that have models of single cells, synapses, and networks. So again, this is my lab in Ribeirão Preto, here in the state of São Paulo. That's the university. Basically, what I do is to study dynamic phenomena in models of neurons and neural networks. And I use both H-type or simplified neurons. So this is an early example from about 15 years ago. In this case, a model of fast synaptic plasticity. So basically, not even facilitation, just this plasticity that occurs when you unblock the magnesium ion from NMDA channel. So you just modeled the increase of synaptic strength when you release the blockage of NMDA channel. So this will lead to an increase of synaptic efficacy. This would be enough to simulate this experimental phenomenon here. So people, I mean, yesterday I mentioned to one student. So you all know that we have in the somatosensory cortex of our brain, and also on the motor cortex, representations of the entire surface of the body. People did that when epilepsy patients allowed their heads to be open for surgery, and then they allow neuroscientists to stick electrodes there and map the response of the patient who is awake and feeling not pain, but feeling the touch of parts of his or her body. And then doing this, Penfield in the 1940s was able to map so-called homunculus, which is a representation of the entire surface of the brain in the part of the, surface of the body in the region of the brain. Here's in the somatosensory cortex. In particular, macaques have a representation of the hand in the somatosensory cortex. So they have representations of the digits. And if you surgically you cut one of the fingers of the animal, what one observes is that the representation, the map, so-called somatotropic map, changes. And so you just cut a digit in the periphery of the body, you don't cut the neurons in the brain. So neurons in the brain that before represented neuron or digit 3, now they, because of lateral inhibition, the phenomenon, the mechanism that I explained yesterday, and synaptic plasticity, they start to represent the neighboring digits, 2 and 4. So there's a new representation in the brain, and you have this amputation. We were able to simulate this using a detailed model of the somatosensory cortex, and we were able to show the same phenomenon. So there's an invasion of the area that before represented, say, digit number 2, which was lesion here, by the other digits. So this is just to show you that it's possible to simulate in a computer model plasticity phenomena. One can also simulate spontaneous activity of cortical networks, cortical-like networks. So networks that capture elements of the cortical networks. And when you are at rest without doing anything, you have so-called spontaneous activity at your brain, and the spontaneous activity is, in general, it displays slow oscillations with low frequency and high amplitude as well, and so-called up and down states. Up state would be a state in which neurons are depolarized, and it's much easier for them to spike than in under resting conditions. So they are in so-called up state. And then when in the cycle, in the oscillatory cycle, they can go to another state, which is the so-called down state, in which they are hyperpolarized. So they change, they keep changing from a depolarized state to a hyperpolarized state, and the resting state would be somehow in between. So neurons, cortical neurons display this phenomenon, oscillation between up and down states with low frequency and high amplitude. And by constructing a random network model, so not really cortical-like, but just random, populated by excitatory and inhibitory cells in the right proportion and modeled by two-dimensional leak integrated fire models like the Zikevich or Ardex that I've shown to you, with the synapses that can change in strength, but not in a plastic way. They just have different values. We were able to, me and collaborators were able to, and also in this model we have a synaptic noise. So we added a synaptic noise term to the synapse. So it's a stochastic model. Could be stochastic if this diffusion term here is non-zero. We were able to construct a kind of a diagram showing all the behavior that the network displays. And you see that different types of behavior. And then we chose one here, this type of behavior that's similar to the one observed in real-life situations under rest. So slow and high amplitude oscillations. And then we studied the role of noise in this situation. And in the noiseless cases, you have up and down oscillations like this. So I don't have time to go into the details, but you have the reference. And then the role of noise is just to break. So weak noise changes from this situation here. So yeah, at least this I have to explain. You have here the up and down states because you see that the voltages have two peaks. It's a bimodal distribution. You have a depolarized state and a hyperpolarized state. So an up and down state, they disappear when you add weak noise. So you basically have oscillations around the resting state. But for moderate noise which is not really large but larger than the weak noise, you recover the oscillatory phase and you also recover the up and down oscillation. So you have, and then but on top of that you have the fluctuations around rest. So you have intermittent changes or jumps between an oscillatory state with up and down states and then quiescent state in which the voltage of the average voltage fluctuates around rest. And then again you go to oscillatory state, and then so you have this multi-stable situation there, which is interesting, very interesting dynamical phenomenon. And then we are studying this as well. Another thing that we are doing, we are using the Porchans-Disman model, the graph that Porchans and Disman constructed. And in collaboration with Disman himself and one of my students, we are populating the model with different types of neurons, not only the leak integrated fire, which was originally used by Porchans and Disman, but by Izikevich and Adex, so more complicated leak integrated fire type models, two-dimensional ones, with different firing patterns. And we are adjusting the parameters to obtain activities in this case, yes, cortical-like model, column-like model, to obtain activities layer-wise and cell-wise that are similar to the ones observed or measured experimentally. So we are tuning the model to obtain firing rates that are similar to firing rates per population, per layer observed in real animals. And then using this model, we are trying to understand how can we generate alpha oscillations, because people observe whenever you close your eyes and if you measure your EEG, you see even slower oscillations than the resting state oscillations. These are called alpha oscillations. So what's the origin of that? So we are studying different mechanisms. In this slide here, I only show the effect of adding intrinsically bursting cells, so cells that respond like in bursting ways, in a bursty way to constant current, like trrr, trrr, trrr. So we put this type of cells in layer 5, and just by doing this, one is able to obtain a peak in the power spectrum here that is close to alpha, 10 hertz. So this is a possible mechanism that would explain why one has alpha oscillations in the brain. One has so intrinsically bursting cells in layer 5. Of course, there should be other mechanisms, but this is at least one mechanism. And this gives you an idea of one type of experiment that can be done in this type of model. So it's like experimental science. You have your own model, which is a setup for you to test different ideas. And then you add some cell type there, and you measure, and you compare to the control situation, and you do all the statistics. and then you add another mechanism, and so on and so forth. So you can really do in silico. You use a computer simulation to do experimental science, experimental theoretical science, because you do experiments there. Another model, just to finish, we are also studying hierarchical modular networks. So we artificially construct hierarchical modular networks using an algorithm that we didn't invent it, but we adapted it to our purposes. So we start with a random network, which has hierarchical level 0. This is the adjacency matrix. And then we split into 2 hierarchical levels with, say, 2 modules each, and then 4 hierarchical modules, and so on and so forth. So you have, these are the adjacency matrix for the types of networks that you can have. So you can have networks with a high degree of hierarchy. This is H equals 3. You can go all the way, depending on how many neurons you have there, you can subdivide your network into different hierarchical levels artificially. But then the idea is just to study the role of hierarchy and modularity and networks. And then we also consider not only the hierarchical level, but also the global synaptic strength. So all neurons are connected by a constant synaptic strength, say J, and then the inhibitory cells are connected by a synaptic strength as J times G, a value that gives the relative strength of the inhibition towards excitation. But J is a parameter that captures the overall synaptic strength of the network. When you have weak coupling and low modularity, you have a behavior like this, very low activity. And as you increase, say, without, say, single random network, by increasing J, you start having so-called heterogeneous behavior. This was well known before we started to work on this. And now we added modularity and hierarchy. And we saw that adding hierarchical and modularity structure to the network also would create heterogeneity in the network. And each module here would start to behave like a single element, single node as well. So there'll be fluctuations in the network. And moreover, we studied how information is transferred. So taking that idea of how structure influences dynamics. So we want to address the question, how is information transfer affected in the network when you have hierarchy and modularity? So there are several possible ways of measuring information transfer. We use the so-called transfer entropy. This is definition from the Wikipedia. T is just from a process X to another process Y is the amount of uncertainty reduced in future values of Y by knowing the past values of X, given that you know Y. So how much you can tell about the future of Y, given that you know the past of Y and X, compared how much you can tell about the future of Y, given that you only know Y. Basically, that's the idea. And then in that paper, we measured transfer entropy based on spike trains of individual neurons, so a pairwise spike trains of all the single neurons in the model. And then we measured the TE here in terms of the hierarchy parameter here and the synaptic coupling there. And you see that the transfer entropy increases with the hierarchical level, so the number of modules in the network, and also with the synaptic strength. More or less, it increases along this line. And then since the network has modules, we took the average activity of each module and then calculated transfer entropy based on the firing rates of the module. So instead of having the spike times of each cell in the model, we took just the average firing rate of each module and did a pairwise measure there for the transfer entropy. And then we found something very interesting. It seems that there's an optimal value of hierarchy. Like in this case, it's 6, only because we had a certain number. There's nothing magic about 6. It's just as an intermediate value of hierarchy from the smallest one you have in your model to the largest one, to the greatest one, for which transfer entropy is maximal. So it's something like that. The flux of information is maximal for a given hierarchical level. So this is interesting. This is a paper that we submitted. Yeah, that's it. And for the ones who are interested in more information about this entire area of computational neuroscience, I organized here in Brazil the Latin American School on Computational Neuroscience, LASCON. That's the page. So this is a bit of propaganda, no? LASCON. It's a 4-week long. So you have time to go out to bars and everything else, to the beach if you stay here for a month. The last edition was in January, from the 6th of January until the 31st of January, full 4 weeks. All students were housed at a hostel in Vila Madalena. Professors were housed in a hotel at Pinheiros. And this is something that I organize biannually, so since 2006, every 2 years. The next one will be in 2022. The structure is more or less similar to this one. You have theoretical classes in the morning, so you have lecturers who are invited from all over the world to teach about, say, single-cell realistic models, simplified models, network models, plasticity models, system-wise models, and so on. And in the afternoon, you have hands-on practical classes. So each student brings his or her own computer. They stay there for the entire afternoon, learning how to simulate using neurosimulators or other tools. And in the evenings, they stay there until 8 p.m. usually. But that's in January, so the days are longer. They work on exercises or project work. So each group of 2 or 3 students have to work on projects, and they have to present orally at the end. There's a full day of oral presentations by students, and eventually some of those presentations, they result into papers as well. So I think nowadays it became a world-class school like this one, but the only difference is that it's longer and it's hands-on. So if you are interested, take a look keep an eye on the next one. And that's the page. Also, I must mention that at the moment I'm part of Neuromat. Neuromat is a research, innovation, and dissemination center for neuromathematics. So neuromathematics could be another name for computational neuroscience from the point of view of mathematicians. I'm not a mathematician, but the mathematician who is the head of this project invited me to join the project. I'm currently the director of innovation. Neuromat is a CEPID, or RIDC, which are projects, special projects funded by FAPESP, the São Paulo Research Foundation. They receive budgets of millions of dollars to do research, innovation, and dissemination on a given subject. Currently, I think there are 19 RIDCs or CEPIDs in the state of São Paulo. Most of them are based at University of São Paulo, some of them at Unicamp, and some of them at UNESP. I don't know if there's some at UNESP. I don't think there are any at UNESP at the moment. But yeah, so the advantage is that you receive a lot of money to do research, and they run for 11 years. So we are already in the second period. They run until 2024. So we have scholarships and positions for students, if you are interested. You can either work here in São Paulo at Mathematics Institute, or at my place in Ribeirão Preto, or at Campinas, because we have researchers from Campinas as well. Actually, we have a whole network of researchers all over the world. We have researchers that collaborate with us from Finland, from Germany, from France, from the United States, from Argentina, from China, from Uruguay, from Chile, so Rio as well, for different states in Brazil, Minas Gerais, and so on and so forth. So you can visit the page to know more about Neuromat. And only 5 minutes late. Thank you for all this opportunity. Questions? Could be over coffee break, no? I guess everybody's tired? No.
In principle, how informative is a connectome? Because you look at something like a Drosophila connectome, but this necessarily is something that varies from organism to organism, right? Because the pattern is some complicated problem of formation of tissue. How
each individual it's a single.
So what exactly is the connect? There is no single fly which has the connectome.
Well, there's the fly that they use to construct the connectome.
Okay, so it comes from one.
Yeah, they select a fly, or they can select say an assemble flies and do the averages, or they can have say different connectomes for different flies and then compare them. But of course, each single individual is different. So eventually, they need to have at least a population of connectomes for a given animal, and then compare, and then have the dynome for them, because the connectome by itself also doesn't help. It's like the genome. We know from the same experience of knowing the genome. It's not enough for us to understand our response to a given disease, for example. We need to understand the dynamics of the elements. So, but it's the first step. Of course, it's a question one can ask. is a positive question. I mean, I want to know the connectivity of the cells within a given system, of the cells in the brain of a given animal. How is it structured? How many, say, and then you can characterize using graph theoretical measures. But it's just a preliminary question. It doesn't help much. It's illusory to believe that the connectome would solve all the problems. But, you know, people have to create those fancy names to get money from the funding agencies and convince the population and the governments that they have to fund us. It's a necessary first step. Obviously, it's not the last one. But it's a necessary step in the development of our understanding about the system.
Well, I see many tired faces. I think we take the discussion to the coffee
Yeah,
break.
let's postpone it.
And I would say we continue around 5:30, provided that Fernando shows up again. Okay. So enjoy your coffee and cake and discussions. Thank you.
Thank you.